Good afternoon, Dr. Sami.
Good afternoon.
Yeah. We'll begin in a short while, sir.
Okay, sure. Yeah.
Yeah. Yes, sir. Sir, we'll start in 2 minutes.
Yeah, sure.
Yes, sir.
Good afternoon, Dr. Sami.
Good afternoon. Good afternoon. Good afternoon.
Welcome to our EFDP online platform.
Thank you.
Yes, madam, you can take over.
Yes, sir. Okay. Over to you, Sonali.
Okay. Good afternoon, everyone, and welcome back. It's my honor to introduce today's eminent speaker, Dr. Sami Mukhaopadhyaya. Currently, he is Principal Application Scientist at Novalet Pharma, Pune. He has completed his PhD from IIT Kharagpur in Synthetic Organometallic Chemistry. He has more than 24 years of industrial experience in which he has served as senior research scientist in synthetic chemical product and process development at Novartis R&D and in computer aided molecular design work at Tech Mahindra R&D. His expertise are computational chemistry, molecular modeling, and material design. He has successfully handled research projects related to novel catalyst design for polymerization and developing strategic alliances with industry and academia. He has been involved in professional activities like exploring business opportunities with pharma and non-pharma industry, client interfacing, and client relationship management. He has also been actively engaged in hands-on training programs in computer-aided molecular design work for the academia, institutes, and industry, and innovation-oriented pharma projects related to the NCE design and novel genotoxicity prediction services for the generic pharma industry. He has several high-impact international publications to his credit. He has also been invited as speaker at various national and international conferences and workshops. So we have with us an organometallic chemist and modern computational modeler for this FDP who is going to address on fundamental aspects of bioinformatics and structural biology and its application. Over to you, sir.
Okay. Good afternoon, everybody. I believe everybody is back with renewed energy after the lunch, and post-lunch sessions usually tend to be a little drowsy. You might have cups of tea, which I'll be missing out because mostly I have been the experience of actually visiting the places and with my colleague Kundan, or there is one more person, Mr. Amit. So this time it's different because of the corona situations and things like that. So we are kind of enforced into doing this kind of a web-based presentation. So the reason for saying all this is that post-lunch sessions, multiple cups of tea. This is what is the usual paradigm when I do the sessions.
Right,
Yeah,
sir. Right, sir.
yeah.
Right,
Not
sir.
only for me but for the audience as well. But I'll try to keep it
Absolutely.
as energetic as possible so that that part can be a little bit avoided. Yeah. So
Yes, sir.
coming to the point, so you see that the title is pretty big because it has got several aspects of the bioinformatics, basic principles of bioinformatics, and typically with a focus to structural biology and its applications. Now, so I'll start with. So this is a little, I think the time is up to 3:30, right?
Yes, sir.
Okay, good. Yeah. So I will start with some of the basic concepts of the pharma R&D, the drug designing aspects, and all that, and then slowly we'll move on to the structural biology, more focused on the structural biology or direct drug design. So if you see the first slide, which I'm showing right now, it is actually nothing but the entire pathway, which starts with identification of the disease and followed by isolation of the protein responsible for the therapeutic area that we are talking about, of course involving genomics, proteomics. These are all very generic terms I'm using here because it involves all of these different multidisciplinary, as we call it, very multidisciplinary applications in this area. So the ultimate aim here is to, of course, isolate the protein on which we are going to work. And after isolation of the protein, there will be several other steps in the process. So there will be, of course, high throughput screening. There'll become, so when I say isolate the protein, once the target is isolated and you know exactly which target to work on. Of course, there are more details like the crystal structures of PDB and all that, which I'll come slowly. But after that, whatever I'm talking about here, screening, combinatorial chemistry, these are all on the ligand, on the ligand molecules that we are talking about. So that at the end of all those filters, I call them as different filters, so that you ultimately try to arrive at lead molecules. Lead molecules is a very common terminology here, which is used in this area. Lead, promising molecule towards a more drug-like candidate. So the ultimate aim is, of course, to find the drug-like candidate by doing all these different kind of screening. So essentially, in a short way, or precise, briefly, if I can say that all these exercises that we do in molecular modeling are nothing but one form or the other of screening or prioritizing. So it could be screening or prioritization of targets, it could be of small molecules, whatever. But the whole thing boils down to essentially finding out which is better than the other candidate. So it's something like that. And of course, once the drug or drug molecules, not just one, but of course we are aiming for the most potent one, but we should have a number of potential candidate molecules, which we can try to make it more realistic by applying several kinds of further filters like ADME, and there are several other pharmacokinetic screens which we might use. And ultimately, when we are happy with all this, we go ahead with the preclinical testing. So that is in short the different steps involved over here. Now, this is actually some of the realities in the pharma industry. Yeah, so this is some of the realities in the pharma industry. You see that this graph very clearly shows from 2000 till 2017, you will find that the new drug approvals, or NDAs as they are known as, you can see that they are more or less somewhat leveling and somewhere increasing in a couple of years and all that. But the important point in this particular slide is the spending. So in the industry, as you know, that there is a very important term which is used, return of investment, ROI. So whatever the company the company is spending, the returns have to somewhat match. And we find here that the gap between the R&D spend, which is the green profile over there, and these actual NDA approvals, the gap is actually increasing. If you see, this is the struggle. That's why you see below it's written there, from wherever we have taken this, the science struggling to keep pace with the innovation. So essentially, this shows that the gap between the actual innovation and the spendings is widening, and that is not a very healthy situation. And that's why in the pharma industry we always keep hearing of this term, that the pipeline is getting more and more dry in spite of having all kinds of tools and techniques and everything else. Now this slide also shows a kind of a paradigm shift. If you again look at from 1999 to 2018, pretty recent, you will find that these red bars are for the chemical entities and the new chemical entities, and these are all approved, by the way, approved molecules, and the yellow one are for the biologics. So as you understand, as you can see very clearly, that the biologics were pretty low in percentage earlier, but they are actually sizably growing. And in 2018, you can see that out of 42 plus 17, if we add them up, we get the total number, that this has almost reached. Yes, it is not exactly half of the small molecular number, but yes, it has sizably grown. So we are trying to show here that the biologics is gaining importance slowly, more and more. Okay, now this is a very interesting slide. It shows that this actually is not a pie chart, but yes, it shows the division of the different categories of molecules that are important. Of course, the largest chunk, as you can see, the largest chunk, 33 molecules. These are 2019 FDA approved small molecules. So the small molecular share is largest. And among the others, antibodies are next, I would say, which is pretty big. And then you have the RNA agents, which are nothing but vaccines. You have the ADCs, which are nothing but, so ADC stands for antibody drug conjugates or complexes. So there are different different types under which we have categorized them. But the largest share is, of course, of the small molecules. So that's what we wanted to show. This is the very latest 2019 statistics. This is an old slide I've been using, I think, from a long number of years, maybe more than 15 years I'm using this slide, because this slide is very interesting and it actually gives you a crux of the whole story, that there are a large number of compounds, large. This could be just representatively I've kept at 10,000. It could be 1 lakh also, 1 billion also. So the important point to appreciate is there is a large number of molecules which we are starting with. And after that, here we are. After that, it's just like a funnel, as if it's a filtration funnel. And this is the entire drug discovery life cycle, as we call it. So this is stage 1, stage 2, etc. Whatever modeling work, etc., that we're going to talk about, or these last few days and remaining days in this particular conference, mostly we are all drug discovery. We are all talking about in the first stage 1, first segment here itself. All the remaining, as you can see, the preclinical tests, the investigation of the new drug submission and approval, and followed by the clinical trials of phase 1, phase 2, phase 3, everything is mandatory, and then the new drug application. So ultimately, after so much of large number of compounds, you might actually land up with one single, if you are lucky, that too if you are lucky. So the thing is that this is a very long process. You can see that total number of years, if you add up, it will be really large, more than 17, 18 years, and billions of dollars pumped in. And still, you saw that earlier slide, that how the gap is there between the spendings and the actual the molecules which are coming out. So because the whole approach earlier was done serendipitously, that means it was done by hit and trial. That was made more rational. So whatever we are studying here in computer-aided drug design, CADD, is rational approach. Rational approach means we are putting in several parameters and trying to filter out which is the more promising lead molecule than the other. So that's why the importance of these drug discovery, different tools and techniques that we are using to achieve that goal. So coming to some basics of bioinformatics. So bioinformatics, rather than basics, I would say what are the different areas it actually encompasses. So you can see that these different components of gene discovery, from gene sequencing and genetic linkage studies and polymorphisms, all of that, that is there because it all starts from the gene level and then it gets translated to the protein or proteomics. So obviously, when you have the genes, you also have these the functions of the genes. So all of that come over here, where the genetic people, geneticists study all of this. We are into this area right now, talking about the drug design, and there you have different techniques of high throughput screening, structure-based drug design, ligand-based drug design, virtual screening, combinatorial library, database searches, and so on and so forth. So, but after that modeling, here comes actually the other things, other aspects. So these are the reality. I always call them as the reality checks. So here you have the clinical trial data, the efficacy, of course, the toxicology, the toxicity, which is a very, very important. It is as important as the efficacy. You cannot just have a very promising, efficacious molecule with a lot of side effects. That will not help. That's why it's a very complex interdisciplinary area. It involves so many people. In this medicinal chemists, organic chemists, synthetic chemists, pharmacologists, a whole lot of people, and of course, in today's paradigm, the computer science people also, who keep talking about AI and machine learning, which is also there in the theme of this conference for you. So the next slide again is kind of the same theme that I was telling you, that the informatics actually involves chemoinformatics, bioinformatics, information technology, artificial intelligence. Of course, this word artificial intelligence has come up recently, quite recently, with the words. It goes with the word smart. So everybody is trying to be smart, but I have my own views on that, which I cannot discuss here. That can be discussed one-to-one. But anyway, the point here, what I'm trying to say is that the informatics has a very strong component of IT, information technology, and several kinds of AI machine learning techniques. Okay, so this is another slide. Again, we have already seen this, but it's a nice slide, nice graphical representation. That's why I have kept it over here. So it starts this is the whole game here. Starts with the target discovery, target identification, validation, assay development, with which you will be testing it in vitro. Then it involves all the informatics that we were talking about. And once you come up with the promising lead molecules, not one, several, let's say, you will go ahead with these the screening part, the assay testing, and all that. You can also develop library, because if you want to design what is known as lead optimization, the modification of the existing active molecules to make them more powerful, more active, that all actually, if you want to do it, in small numbers, it is not very promising. If you want to do on large numbers, then you can develop libraries. So this is very important actually. So this falls under the combinatorial library development, and just library development is not enough. You should also screen them with realistic filters, the kind of filters of virtual screening which I talked about a little while back. Then of course you have to see its activity. You have to against you can do it against cell lines, enzymatic assays, and so on and so forth. Then you can also you also have the disease models, and then you have to look at, it's very important, is the mechanism of action, because the whole of this actually rests on the mechanism of action. What is actually the mode of binding? What are the, for example, in docking, I'll be talking about a little later, what are the different interactions which stabilize that particular ligand protein complex, let's say, for example, as an example. Okay. Then in the preclinical development, you have all these different components. So you have pharmacokinetics, in vivo pharmacology, toxicity, safety assessments, and all those kind of things. So basically, you can see that this game is very complex, and it is pretty multifunctional or multidisciplinary. Okay. Now coming to specifics of computer-aided drug design, CADD approaches. So there are two approaches. On the right side, you have the usual ligand-based approach, which is also known as indirect drug design, and on the left side, you have the structure-based. Structure here means structure of the protein, the target. So this is the SBDD and this is the LBDD, as they're abbreviated. So in the structure-based drug design, you can have scenarios where the protein structure is available, the crystal structure of the protein is available, and in another scenario, the crystal structure is not available, as all of you know that a major chunk of the protein target, the crystal structure is not available. And of course, 10 years or 15 years back, when we were doing and speaking on this subject, we used to say GPCRs is a classic example of that, but now it is no more the same. GPCRs also, there have been some, due to the advancements of technology and other things, there have been some structures which are available now, but still there is a major section of GPCRs, ion channels. So these are, different systems where the crystal structures are not so readily available, where you follow the homology modeling path. And the homology modeling path, all of us know, is a pretty long path. It takes a lot of time. The accuracy level is not always that great. Sometimes you might have to do threading. All those things are challenges in homology modeling, but you can't help it. If you don't have a crystal structure, you have to take resort to that. But ultimately, whether you have a crystal structure or you develop a homology model and validate that using Ramachandran plots and other things, ultimately you have to study from both, whether it's a crystal structure available or you do a homology model and validate the structure and see whether it's a good structure or not. You ultimately have to study the protein ligand interactions. This is the most important point over here, because this is what I was saying, that when you want to study the mechanism of binding of these different potential molecules, lead molecules to the target, you need to see what are the binding modes, what are the binding interactions that stabilize them. And once you have got a good feel of the protein ligand interaction, binding interaction that stabilize that particular complex or assembly, you can actually go for modification of the ligands within the active site. This is nothing but lead optimization that I was talking about. Lead optimization would come in QSAR, it would come in docking, everywhere it would come. Ultimately, it means that you are trying to modify the best molecule in your data set and try to achieve at better than the best. I call it that way. On the right side, you have the ligand-based approach, which is nothing but the indirect drug design. So in the ligand-based approach, you have the QSARs. You have the 2D, 3D QSAR. It can go up to ND. But right now, I'm talking about the most commonly used 2D and 3D. And let me tell you from an industry perspective, because I have got a experience of several years of industry projects working with all kinds of industries that you can think about. So that's the reason why I always call this area not as CADD, computer-aided drug design. Actually, I call it more as computer-aided molecular design. I'll tell you why. See, the point here is we are trying to come up with a novel molecule for some novel application. It may not be all the time pharma. If it is pharma, yes, it is drug design. We have equally have experience of working with the non-pharma sector. So I'm talking about the beauty care and the lifestyle and beauty care companies. So we have worked with the top-notch companies in the world in this using informatic techniques. So we have worked with the Unilevers, with the Procter & Gamble's, with L'Oreal's, everyone we have worked with in different projects in the last the number of years that I'm here in this company. And apart from that, we have also worked with agrochemicals, like ITC, for example, whose main business originally was agro. So what I'm trying to say is the verticals or the industry applications might differ. It is not always only drug. So more holistically, it should be called as computer-aided molecular design. That molecule could be a novel molecule for XYZ applications. But coming back to our current topic, as I was saying, that why I deviated a bit is for these kind of applications when we are talking about the non-pharma sector, whether it's an agro or a petrochemical, Reliance. For example, we worked a long time with Reliance Industries for catalyst design for their Hazira plant, where they have polyethylene and polypropylene and all of that. That's actually my organometallic experience directly put to use from my PhD. So what I was saying is, in these kind of areas, non-pharma areas, the ligand-based drug design that I'm talking, and this is important for this audience, whoever is listening to me today, because don't get that, bogged down by the idea these are only applied only for drug design. In fact, depending on the application, you should know. This is my famous statement. I keep using it everywhere. You should be smart enough. This is real smartness. That you should be smart enough to know what to use where. This is very important. What to use means what tools to use. Where means in what application. In case of the non-pharma sector, this with our industrial experience, I'm telling you, with several years of almost 2 decades of projects that we have done, this area is very, very important for the beauty care, lifestyle people, for the agrochemical people, for the these polymer people, plastics industry. This area is very important. You know why? Because simple example I'll tell you. In the case of skincare products that all of these big guys are using globally, the target tyrosinase is extremely important. Just a little bit deviation from, because I wanted to bring this industrial flavor. It is not just I'll run through the slides and that's the end of it. You should also understand that how it is applied in the real life in the industry. In the case of tyrosinase, is a very important target. Tyrosinase we have worked extensively with different non-pharma companies. Now, tyrosinase, the crystal structure was not available till very, very recently. Okay. Now, therefore, major part of the work that we did on tyrosinase was, of course, different variants of tyrosinase was mostly done using these kind of QSR techniques. And that too, let me tell you, 2D QSR, not even 3D, because 3D is a very challenging. There you have to align the molecules in 3D, and there are so many prerequisites. Industry doesn't have the time for it. There is a certain amount of time which has been given to you with a deadline and a certain amount of money that you're getting for that project. So these are very important parameters that you should keep in mind. At the end of the day, for an industrial delivery, whether it gives you a publication or not is not the main goal. The main goal there is to deliver what you have been requested for and what you're getting paid for. So this is a very different scenario. And in that scenario, you should know quickly to use, smartly to use what techniques. That's why I said what to use where. And let me tell you, just for your information, these kind of ligand-based drug design, it is not just QSR alone. It is QSR, more I would say 2D QSR, because that is faster. It is combinatorial library. It is database searches, chemoinformatics, virtual screening, pharmacophore modeling. These are the ones which are more used. So all of these, as you can see, are nothing but ligand-based drug design. These are not actually structure-based because you don't have the structure. Tyrosinase, for example, is extremely complex. You don't have the structure. The structure came in very, very recently. And so I mean, just in this slide itself, I wanted to tell you a flavor of how the real life goes, how the industry goes. I mean, what kind of tools you use for what kind of applications, what to use where. That's why I said. Anyway, coming back to this slide again, that when you use these, when you do a QSAR or you do a library, combinatorial library, you need to put in realistic filters, blood-brain barrier permeation, toxicity predictions, solubility, so many real-life virtual screens or filters you need to put so that at the end of the day, you arrive at most potential lead molecules, and which you can take up, of course, for the preclinical trial. Okay, a little bit faster now. So now this is more details of the direct or target. This all is, I'm sure it's known to you. So I'll go a bit fast because it's already close to 2:30, and I would like to speed up. There's a lot of slides actually on docking and other things. So let me little bit speed up on these slides. So you know all of this. This is like further details that if you have a crystal structure, what you do. If you don't have a crystal structure, what you do. I already told that you need to do homology modeling. In that case, you don't have a crystal structure, model validation, and using Ramachandran plots and many other checks and balances, optimize the the protein that you've homology modeled. And ultimately, at the end of the day, again, as I said, that you perform the binding mode, the binding studies, whether it's a homology model or a crystal structure downloaded from the PDB, ultimately you need to do ligand binding studies, and based on the known ligands, you try to modify and come up with more promising lead molecules, and ultimately go ahead with synthesis. Now, this particular pathway, again, as I'm telling you, is a little old concept. Nowadays, once again, I'll tell you, in the industry, pharma or non-pharma, more of non-pharma, but also in the pharma, people are nowadays no more actually following this conventional path of synthesis and all, because it takes a lot of time, and at the end of the day, as you know, synthetic chemistry is no joke. And so the thing is that instead of going for synthesis, if you can modify the ligands and get a hang of the pharmacophore of the designed ligands that you've developed after doing lead optimization, and use that pharmacophore to search known databases. Like, there are so many chemical databases available: PubChem, ChemBL, BindingDB, okay, Drug Bank. There are several good quality databases. Zinc, for example. These are all very well known databases. If you can use that pharmacophore, after lead modification, you take your ligand molecules, develop the pharmacophore, use the pharmacophore to search against chemical databases, come up with new scaffolds in that database search, and those molecules are and you should be focusing also, while doing that database search, this is again from industry perspective, when you're doing the database search, you should be actually focusing on what kind of molecules are readily available with the vendors. So you see how the paradigm shifts. It is no more the age-old concept of doing all this and then go to the lab, synthesize them. You see that they are not good for synthesis. It wastes a lot of time. Today, the people, whether pharma, non-pharma, more of non-pharma, but also the pharma, once the modeling part is over, they want to do a quick search and zero down on molecules which are already available with the vendors. Because when you are doing a scaffold hopping, as you know, that on a property-based search from different databases that I told you just now, you would actually land up with novel scaffolds for that particular application or therapeutic use. Now, those kind of molecules, if they're readily available, that also is a criteria when you filter out the molecules from the database, which are readily available, let's say, which are commercially available, which are cheaper. So based on that, you can now go and apply, procure them from Sigma, Aldrich, whoever you want, and then get those directly. So you can understand that how much time is saved avoiding the laboratory synthesis. So those are known molecules, but for a different application, and so you buy them. You get them, buy them from the chemical companies, and you start directly with your in vitro test, cell line testing, and all of that. So, to cut a long story short, there have been lot of changes in the way even molecular modeling is done nowadays, and particularly with an industry perspective. That's the reason why I'm telling all of this with each slide. It is not just going through the slide and that's the end of it. I'm trying to bring in how the industry works when these are applied in the real life. Okay. Now, structure prediction. So here we are talking about homology modeling. So if you can see very well, homology, as all of you know, means comparative modeling. So there is a sequence similarity, and so you have a target whose crystal structure is not known, sequence is known, and you know very well that it lies on the principle that if the sequence is similar, then the structure should also be similar. So that's on that. That's the principle. So usual homology modeling is done when all of us know that between the template and the target, if the identity is more than 30%, then usually 30%, 35%, 40%, then the usual methods of homology modeling would work. But if it is less than 30%, 30% is a empirical cutoff, you can say, because with lots of data sets, it has been seen. It is lower than 30% identity between the template and the target. I'm sure you know all these terms. Then after performing BLAST, of course, then one has to do much more rigorous homology modeling, which is known as, it's not really the usual homology modeling, it's called threading. Okay, so it's much more complex, takes more time. And if you want to deviate from all of this and don't want to look at the similarity or identity between the template and the target sequences, then you can go for ab initio, which is even more time consuming, and of course it has its own rigorous and accuracy levels, but it's very, very time consuming. But people, as a modeler, you should know again what to apply, where to apply. Important. Now, in case of protein modeling, these are some just pictures, typical snapshots, where you see that from the DNA you are moving on to the translate to the protein, and then so in amino acids, and then you have the 1D structure, 2D structure, 3D structure. This is the actual 3D. This is how it looks like in in our own software. We have a software which Dr. Bhatia is well aware of, and many of you may be also aware of, called VLife MDS. These are all snapshots taken from our software. So this is how the 3D structure looks like. And this is another snapshot where there's a ligand docked inside the active site, and this is the active site studied with more details in terms of electrostatic potential and all of that. I mean, basically, these are all just snapshots of docking studies of ligand molecules inside the active site. Now, in case of docking, this is a cartoon diagram. This is the receptor, and this is the ligand. And the so you can see very clearly that there is a pocket, the cavity, and the ligand goes and sits over there. So ultimately, this is the receptor R, this is the ligand L, this is the complex RL, and this is the association formation, and this is dissociation, as you know very well. And this is standard formulae. All of you know that actually. Now here, so why I'm saying all of this is that from this equilibria, actually these free energy equations are derived. These are all very well known. But what I'm trying to draw your attention is this delta G, free energy of binding between the protein and the ligand, has got an enthalpy term and it has got an entropy term. So the point is that whenever we do docking and we calculate these binding energies, we mostly are on the enthalpy. We don't really go for the free energy unless and until it is required. For more accurate calculations, you have to bring in the entropy factor and then calculate the free energy of binding. So why I'm showing all of this is that these are the equations that are involved when one wants to calculate the binding energy between the protein and the ligand, free energy of binding. Otherwise, we are happy with the enthalpy part. And because ultimately, at the end of the day, there are some, you can say it's philosophically speaking, but philosophically speaking, I told you right in the beginning that in modeling, we are trying to just compare and prioritize or rank, order, or screen and say these are better molecules than the others. So let us look only at the better molecules. So it's a screening exercise. It's a prioritization exercise. It's a comparing exercise. In a comparing exercise, we are not really interested in absolute accurate values. Even if there's an error, that error will be across all the molecules and we can still go ahead and compare and get the trends. That is the reason why if enthalpy itself helps me, I won't really go for entropy inclusion and making life more miserable. Already my life is very miserable. So if you want to make it more miserable by bringing more accurate terms, try to bring in more accurate terms. But I'll show you a little while ahead that I need to speed up because I need to get to those slides and show you how docking has its own problem. So nothing is, a piece of cake. When you are given a piece of cake, you remember there are also some, thorns inside. Anyway, this is my favorite slide. I'm using it for, again, the last how many years, I don't know. But I like this because this is a mother and child, what shall I say, association. And according to me, that mother and child association is the most noble, most sacred, the most beloved association that one can have. In fact, with parents, but particularly with mother. And that's why it's a very favorite slide of me. So this is what it means that whenever ultimately you're doing docking, if this is the mother is the protein and ligand is the child, ultimately we are trying to see how much the interaction between the ligand and the protein is maximized. Of course, I'm talking about favorable interactions here, which will lead to minimization of the total energy of the complex protein ligand complex. This is the crux of docking. Try to get those poses or 3D orientations of or placements of the ligand inside, which will give you the maximum possible hug. This is a real hug of mother and child. So how much the protein and the ligand hugs each other, that means basically how stable that entire complex is. That depends on how much favorable interactions are maximized in the process. Now, coming to more bland things which need to be covered. So in this process of docking, these are the steps. In the receptor preparation, we all, I'm sure you must have used software, different software, and you know these are the steps. So I'm not getting into those details, but you know that very well. So if you get the crystal structure from the PDB, if you are lucky enough to have the crystal structure from PDB, you can download that and do all of this, which is nothing but protein preparation. Similarly, you also have ligand preparation where you draw them, optimize them using some energy minimizer. You develop the conformers. If there are protonation sites, tautomers, you take care of all of this. So basically, this is like ligand preparation, and then ultimately you see how nicely they dock. So in case of docking, we are actually trying to see what is the, we are trying to rather not only see, we are trying to first we are trying to establish what is already known, the known ligands with the active site, and in terms of the binding interactions and all of that I told you just now, best possible interactions, not always the score, but interactions. That's very important. And after that, we can use if we are confident about the binding interactions of known ligands against a known active site, then we will go ahead and try to predict what shall I say, similar molecules that could bind similarly with the known molecules that we have already studied. So here there are two very important terms in docking. One is called the placement, and the one is the placement. That means the 3D placement of the ligand inside the active site, and the second, which is also known as the pose. And the second one is whether it's a good pose or a bad pose. That means whether the interaction is favorable interactions are more or not, better or not, is by a scoring function. Scoring function is nothing but a summation of certain energy terms of interaction between the protein and the ligand. Now, whenever we talk about ligand placement methods in docking, there are some systematic methods and there are some random methods. So systematic is that you will have a grid. At every grid point, you place the ligand, you rotate it by a step size of rotation, calculate the scores, and you try to zero down on the best possible, minimum possible score with the best pose. Now, there are some also random pose generation methods like generic algorithms, random forest, and others. These are some fragment-based methods. Here you can very well see this is a typical ligand. You have fragmented the ligand into different parts. Each part you are trying. This is the active site cartoon. In the active site, in different parts of the active site, you are trying to see based on shape, size parameters what could be the best possible fit. And if you know each of these fragments fit at those particular portions, then you immediately get the best possible orientation of the pose inside. This is one way of doing it actually. Fragment-based approach. This is a little advanced, so I will not really get into this slide with all details, but I will tell you the crux of the story. I'm also keeping a tab on the time. It's almost 2:40, so I'm going a bit. I am a bit slow, but I hope. By the way, am I fully audible? Everything is fine.
Yes, sir. Everything is fine.
Good. Yeah.
Complete
Because
yeah, audible.
Yeah, silent. So I just wanted to do a check in between. Yeah. So what
no, everything is going fine, sir.
Good. So what I'm saying is this particular slide is a little advanced, but all I'm trying to say is that in case of, so there's a term called ensemble docking, which means that you have, see, in the real life, it is not just one protein and a set of ligands. It is a set of proteins and a set of ligands. For example, let's say you're working with kinases, AKT target, AKT1, AKT2, AKT3. These are all the subtypes of families. Now you take one of them, let's say AKT2, AKT1, let's say. In AKT1, there might be several PDBs available on the PDB. Now, when you are studying that, so you can take all the subtypes or all the PDBs within the AKT1 family and then the corresponding ligands, and you want to dock. So now you have a set of targets and a set of ligands. So ensemble docking involves set of ligands, set of targets. But this can be done using, you can use batch process, whatever you want, and then you have to introduce protein flexibility also. It's computationally quite intensive and it's pretty costly. Instead of doing that, there is a particular method called consensus-based induced fit docking. It means nothing but just see those, study those, compare those different family members within the particular subtype. Let's say AKT1 as an example. Just compare them, compare their active sites. Okay, what is the similarity between them? You can overlap those sites. See, you can do a lot of molecular modeling, techniques, and see and compare those and try to come up with a consensus active site by which actually involves all of those different individual active sites of the target. Similarly, also for the ligands that you want to dock. So essentially, from the target family subtypes, you come up with a consensus active site or active sites, and similarly also a consensus ligand from the set of ligands which are known to bind to that those targets. So in other words, both the consensus cavity and the consensus ligand that you come are virtual. They do not really exist, but it is like, you can say, representatives. They're representative of the targets and representative of the ligands. So you can do, then the docking between these two would be much less cost, involving less, the competition cost would go down essentially, because you are actually essentially docking one consensus active site or maybe two or whatever. One consensus, you can always rank the consensus active sites. But all I'm trying to say, this is a way to reduce the computational efforts and therefore the corresponding costs also. It's a little advanced, but I just wanted to quickly brush through that slide to give you an idea of how people do this. Anyway, coming back to scoring functions. So now, scoring functions, as I said, is a mathematical term. It consists of several energy terms, which are nothing but interaction terms between the ligand and the protein. What kind of interaction terms we'll come to a little later. Now, it's a mathematical term, as you can understand. The purpose is, during docking process, they serve as a fitness, how good the ligand is fitting inside the cavity, how good is the pose. And so they can obviously be used to rank. I told you right in the beginning, here the entire game is how to differentiate between the better candidates and the poorer candidates. So here we are trying to prioritize or rank the ligands by using the scoring functions. So if you develop different kind of scoring functions with different scoring terms, you can apply to several protein ligand systems. It is not that one scoring function is only for one given protein ligand. You can apply it across different protein ligands, but it has its own challenges. I'll come to that a little later. But it requires the different information of the poses of the 3D poses, or 3D orientation of the ligand inside the cavity. That is very, very important. These are the different types actually. There is a force field type, and there is empirical and knowledge based. These are the different scoring function types. Force field, as you can understand very clearly, that the interaction terms between the protein and the ligand, these are calculated by different force fields. So steric term, electrostatic term, and all of that, solvation, desolvation, etc., etc. Empirical, as the name suggests, empirical is, so I'll tell you, empirical, if you want to clearly understand, it is just like QSAR. What is done here is that if you know the binding constant of set of ligands, let's say we are studying one target and a set of ligands, if the corresponding experimental binding constants are known, and you know very well that the binding of the ligand and the protein, or binding constants, can be experimentally derived also, and there are some calorimeters for doing that, specialized calorimeters. So if you have the experimental data of the binding energy of binding, or binding affinity between a set of ligands, and let's say to make life simple, I've said one particular target of my interest, then what we will do is that we will take it of like the activity of QSAR and the interactions between the ligand and the protein, which I told you just now, which could be hydrophobic, hydrogen bonding, electrostatic, you know all of that. So all those interaction terms between the ligand and the protein, which actually gives rise to binding, would be nothing but the properties or the descriptors. And we can fit just like it is a QSAR, in fact, where we do a QSAR model where the binding experimental binding affinities, or binding affinities, is fitted to the properties which are nothing but the binding interaction terms between the protein and the ligand. This is the crux of empirical scoring functions. And obviously those binding interaction terms would come up in the scoring function itself of your choice. Knowledge based also is somewhat like that, but it's a bit different, because in case of knowledge based approach, we are not taking binding affinity. Instead of binding affinity, we are taking the actual distances between the protein and ligand atoms, as in the crystal structure. These are actually defined further as we go ahead. But right now, since I have talked so much about the binding interactions between the protein and ligand, you can see this slide. This shows that as the level of, so when you bring in more and more interaction terms, contacts, favorable contacts, unfavorable contacts, Van der Waals interaction, hydrogen bonding, electrostatic, hydrophobic, and so on and so forth. And then entropy of desolvation, it is marked in red, because all the rest of these terms, we are trying to see how much they stabilize the system. But in case of desolvation, when you remove the solvent, which is typically water molecule, in case of from a crystal structure, actually it leads to kind of little bit of destabilization. That's why it is marked in red. But the point in this slide is that as you make more and more scoring terms and try to make the scoring function more and more accurate, of course the level of approximation goes down, level of accuracy goes up, but so is the time. So that's why you will have, I'm sure you know, single precision and all of that, which Schrodinger uses, used to use. I don't keep nowadays track, but I'm sure single precision and multi precision, all those things. That means scoring function is getting more and more refined in order to get a better picture, but at the cost of time, because you're adding more terms to it. That's why. Now, there's a lot of mathematics. I won't get into the mathematics, but just to tell you, just representatively how these look like from some of the most popular scoring functions like autodoc. These are force field scoring functions, and they use the amber force field. These are the terms. This is the typical Lennard-Jones R6, R12 Van der Waals term. These are the protein ligand interactions. As you can see, these are the electrostatic components over here. And then within curly brackets, you have these three terms, which are termed as summation over ligand. This is nothing but the E internal. These are terms which are within the ligand itself. Just like intramolecular and intermolecular hydrogen bonding, these are the intra-ligand terms. That means these are steric interactions, electrostatic interaction within the ligand itself, not the ligand protein. Whereas these are the ligand protein complex terms of similar. So these are how this is how it looks like. Just to give an idea, a representative equation. Similarly, there is another force field scoring function, D score. I'm not getting into these things. Similar, just to give an idea. Then there is this very famous G score used in Gold and Sybil programs, where again, you can see similar terms, but you can see that in the hydrogen bonding term, there are some terms DA, donor acceptor, WW, water. So they have actually introduced solvent water and to make it more realistic, and the hydrogen bond mediated by water. So they have introduced more realistic. That's why I said we can introduce more terms for making it a more and more what shall I say, sensitive or accurate scoring function, but at the cost of time. Now more. So I'll not really get into all this. Just to give you an idea, I told you this is a very so now so all these earlier were force field scoring functions. Now I'm moving over to empirical scoring function, and this is one example. Actually, this Lick score also was an empirical scoring function, but instead of Lick score, I am taking this a little bit more in details. I'll tell you, because the reason is this PLP, pairwise linear, piecewise linear pairwise potential. So this PLP scoring function is a very, very simple empirical scoring function. It has got only 3 terms: hydrogen bonding terms attraction, hydrogen bonding terms repulsion. So this is donor acceptor, acceptor donor, donor donor, acceptor acceptor, and this is dispersion or the steric term. So in empirical score, this is a typical graph. You will find whether it's hydrogen bonding or whether it's steric, the graph will be very similar. Here, this R is nothing but the distance between the protein and ligand individual atoms, and this ABCD are nothing but, I told you a little while back that this empirical approach is nothing but just like QSAR. So when you fit, as I told you, when you fit the experimental binding affinity or binding energy between the protein and the ligand with the properties, in this particular case, the properties would be hydrogen bond, the steric term. These are the different interaction terms in the scoring function. When you use them as properties or descriptors and build up a QSR equation, then the best possible QSR equation would have these constants A, B, C, D. So, for example, and by the way, these are all arbitrary units. This is not angstrom, arbitrary units. So the fact that this is the highest energy that was around 20, all arbitrary units. This is the minimum E -2, and these terms, the ABCD constants, the E and F, these are all derived from the best possible QSR model, because this is nothing but actually QSR. Okay, so that's how, and I'm spending some time on PLP because it's a very simple scoring function, really very simple. It has only 3 terms, but through hydrogen, the polarity of in the interaction between the so the charge interactions I'm trying to say is taken care of somewhat by the hydrogen bonding term itself. So that's why I'm saying that without much complexity, this particular scoring term, it is there actually in our software also, in our docking method called GRIP, and it gives pretty good results. It gives us pretty good results. It's simple. We should keep things simple. If they are simple and still give us the good trends, we are happy with it. That's why I spent some time with this PLP scoring function. Knowledge-based scoring function, I won't get into the details of this slide because it's not really required. But what is important is that, if you just forget about those equations, what is important is that here, as I said, that instead of the binding energy, what we are trying to do here is, see, we have written over here, instead of binding energies, we are trying to fit the experimental data or X-ray data of the protein ligand distances of atoms between the protein and the ligand. That is the experimental data, which is fitted with the different the properties. So this is how this particular approach is. Now we are coming to some more important approaches. So there's a term called consensus scoring function. So what it actually means, I will tell you. What happens is that there's a major problem with scoring functions. The point is that I already told you, scoring function is just a mathematical number. It's actually nothing but sum of certain energy terms. The problem is that you cannot have a single scoring function that will work with all known protein ligand assemblies or complexes that you can handle. So since one single scoring function, there is no what's the right word to use? Omnipresent, omniscient scoring function that can apply for all protein ligand complexes. Okay, this is a major problem. There are many problems. One of them is this. So that's the reason why you are given a choice of different scoring functions for different protein ligand systems, and you have to try and see which one works better for what kind of protein ligand system you are working on. That's why I said you should know as a skilled modeler what to use where. That means what scoring function to use for which scenario, which protein target you are talking about working on is your choice as an expert modeler. So because of the problem that you don't have a single scoring function that can work with all the proteins that are known and their corresponding ligands, therefore people have tried to come up with consensus scoring function, which is nothing but, very simply if I put it, it is nothing but an algebraic sum of the linear combination of different kind of scoring functions. So there is, for example, C score, which combines D score, PM score. So these are the, and in that combination, nobody is preventing you to take. You can take in the combination, as you can see also. For example, in C score, you can very well see there are some empirical, there are some force field derived scoring functions. There are some empirical scoring functions. Similarly, cons doc, which is used in case of it's a combination of doc, flexex, and gold. All I'm trying to say is you can take different linear combination of known scoring functions and try to come up to consensus scoring function, which is supposed to be more accurate than the individual scoring function. But the problem here is, how would you combine them? That is the whole challenge. There are different ways. Algebraically, when you are combining them, you can take different linear combinations. You can use all the operators that you know, but which one works best for a given protein ligand system is, you have to find that out. It's not a one-shot exercise. It is again iterative. It is a challenge. So in case of scoring functions, this is the scenario actually. So it's just a cartoon diagram that after the lecture is over, I'm asking how much you understood. So they're all saying, yes, yes, you have got the point. But actually nodded saying that we have got the point. But inside they're thinking that, this part I have not understood, that part I have not understood. So getting to a global consensus scoring function is as challenging as trying to get an idea across to a big section of people. It's just a cartoon diagram, that's all. Now, there are some examples which I've given, which I am not getting into right now because of the time factor, because there are still a lot of things to cover. So what I'm trying to say is, these are some examples. These are some examples. I've just quoted the corresponding publication also. 11, they have studied across 100 protein ligand complexes, 11 scoring functions they have actually tried to take, and they have compared them and also tried to take a consensus, and they see that it actually predicts better. So I will just give me 1 minute. Yeah. So this is the main point, that the consensus scoring function gives much better prediction of the experimental pose compared to the individual scoring functions that have been used in that. Okay. Now this is another such example which I'm not getting into. Anyway, there are several examples. I've just taken. Yeah, but here there is something interesting. I can tell you that here. See, for example, here in this particular work, by the way, they have taken the gold score, they have taken the glide score, and all of that. Ultimately, they have individually, when they try to compare, I think they got 70% accuracy. But when they took a consensus scoring function of these different scoring terms, they got much more, 80% or more, as the success rate, or the how much it is matching with the experimental data. So the crux of the story, as I told you just now, there is no single scoring function which is capable of working across all the different protein ligand systems. Since we are at a good enough stage to share with you a slide which shows all right from 1981 till 2011, what are some of the examples? These are not comprehensive but representative. It shows some of the examples of these different drug molecules that have come up through structure-based drug design from different companies altogether. And this hepatitis C, this is one of the latest 2000. I mean, not really latest in my slide. It is the latest. Okay. Now we come to further things, a little bit advanced things. So the thing is that, as I told you, that in case of docking, there are two very important terms. I already told you that you need to study the binding interaction between the ligand and the protein in terms of the scoring function, but that is just a mathematical number. More important is the interaction, stabilizing favorable interaction between the ligand and the protein. Apart from that, the accuracy of the docking method can also be checked by the RMSD with respect to the native ligand. So if your protein has got a co-crystallized ligand sitting inside the active site, which is also known as the native ligand, it may or may not be there. If it is there, then you can take it out, you can extract it out, you can energy minimize it, develop its conformers. Okay, that means you really disturb it or perturb it, and then you redock those conformers into the active site, and try to see in that docking what is the best possible pose that you can come up with, which is closest to the original. You already have the original conformer of the native ligand, how close it is. Obviously, you can understand the lower the RMSD, the better it is. Now, this kind of activity, as well as if you want to do some virtual screening of active molecules from searched from different databases using whatever pharmacophore-based search, whatever you can do. You can employ this database, which is known as DUD. DUD is standing for Directory of Useful Decoys. What happens is there are some 40 diverse set of protein targets over here with their corresponding active molecules, as well as a set of decoys. Decoys mean these are molecules which are not for a given target. These that set of decoys means they will not bind to that target nicely. They will be poor binders, so they will have that's why they're called decoys. They are poor binders to those. So in this particular database, for each of those 40 targets, the set of actives is also given, the set of decoys are also given. So what happens is that whenever you are supposing you are trying to test, you have come up with a new docking method and you want to test that, then what you can do is, just now as I told you, if you have a co-crystallized ligand, extract it out, you energy minimize, develop its conformers, then let's say you've chosen one target out of this 40 and you have done this exercise. Now the conformers which you have developed of the native ligand, that together with the active compounds for that particular target, everything, and the original native ligand, everything, you take it as a ligand set and dock. And when you get the results, you can see if your docking method is accurate. You try it, of course, you do the docking several times and try to come up with the best possible results, and then you see that how much your docking method is accurate in terms of picking up the original pose or the conformer of the native ligand. That means which one has got the least RMSD of the among the different conformers of the native ligand which you dock. You can compare them, and this actually is one way of finding out the accuracy of your docking method. So that's the reason why I told, when you are developing a new method of docking, these become very, very important. We have a method called, in our software in VLab MDS, we have a method called the method is called grip, GRIP, grip. And in that method, the scoring function is PLP. And when we developed that method, that was almost 15 years back. That time we did all these exercises, because when we come up with a new algorithm for docking, and then we need to really test it across different data sets. We tested across a huge number of, we tested, I think the entire drug bank was also, most of that was utilized. But the point is that we also took the help of DUD, this particular database, because you have to see how accurate your docking method can become, so that it is able to distinguish, or we call that as, this term called cherry picking. So how much it can cherry pick, that means it can differentiate and pick up the right candidate molecule. This is cherry picking. Okay, now limitations of docking. So docking is not the end of the world. Unlike, and already I tried to give you a flavor. Dr. Bhatia, are you still around? Dr. Bhatia, are you still there? Hello?
Hello, sir.
Yes, sir.
Yeah, Dr. Bhatia is still there? No problem. I don't have to call. I just wanted to know. Yeah.
Yes, sir, he is there.
Okay, no problem. Yeah, okay. No, because this is like, I'm talking and talking, so I just wanted to check it out. Okay, so so we are at a stage now, after speaking of so many different aspects, we are at a stage now where we are talking about the limitations of docking. Why I said this, because I understand this is a faculty group of people, audience. So I'm telling you again, because it's just around 3 o'clock, I still have half an hour. So that's why a little bit, again, I'm taking some real life examples. See, docking, I have seen over my last 24 and half plus years of working in this area. Of course, the first 3-4 years was in Novartis as a synthetic part, but post that, let's say 20 years, I've seen one thing. The pharma world, the industry I'm talking about, and that too the drug discovery companies, which are more and more shrinking in number. But anyway, they are absolutely obsessed or biased to docking. The reason is that somehow, I'll tell you the reason also, because I have really hobnobbed with these people, so both the pharma and the non-pharma. And so the reason why they are so biased, and the whole bias has come up because in docking, you have really, you can get a feel of the protein inside the active site, the interactions. Somehow the feel is better rather than just statistical numbers in case of QSAR, for example. So the ligand-based part is a bit neglected in the pharma world, let me tell you very frankly, but it is not so in the non-pharma world. So when I talked about the FMCG, all these beauty care lifestyle product companies, and the others. So all the Colgate Palmolives and these Unilevers and the Procter and Gambles. So these people, skincare people, and then others, all these people, for them actually it really doesn't matter. To be frank with you, they are not obsessed so much with docking. One of the reasons is also that in their case, just as I took the example of tyrosinase, because that is one of the most common target that is being used for all the skincare people. uh, the point is that the target crystal structure is not available. So there, the game is more of the ligand-based approaches. So that's why I said, why I'm going back to that discussion again, is that docking has its own limitations. If the crystal structure is not available, and it's a very challenging exercise to do homology modeling on a target like, for example, tyrosinase, then what other way, best way you can do other than ligand-based drug design, for example? Because in ligand-based drug design, you do not really take the help of the target right away. The target is implicitly there. In QSAR, it is there. No, I'm not saying it is not there. It is there through the activities themselves, or through the properties that you're going to, because those properties are, represent the protein ligand binding, for example. And you know very well that QSAR can be applied to a set of molecules which have the same mechanism of action or mode of binding to the. So it's all there, but it is implicit. It is not explicit, as in docking. So all I'm trying to say is that, of course, you should know what to use where, but the point is that in case of the non-pharma applications, the ligand-based applications are extremely popular, and they really give us very good results, because simply because that the crystal structure of many of the targets which are important for the non-pharma industries, non-pharma sector, there, because the crystal structure is not there, and really people don't have the time to develop a fantastic homology model, because the bus will go away if you insist. So you have to understand what is the priority order over there. So with that context, or with that background, I'm coming back to the slide that docking is not the end of the world, unlike the way the drug discovery pharma industry people, I'm very specific about what I'm talking about. The pharma industry drug discovery groups are really at one point of time, now the little bit changes have happened, but still it is not the way it should be. So they are very much obsessed with docking. But docking also has its challenges. What are those challenges? Just a few of those challenges. One challenge I already have talked about, that the scoring function, there is no global scoring function across different protein ligand targets. In fact, if you allow me to say, in 2010 or 11, I've forgotten, maybe it was 11, in all the IITs, I'm from IIT Kharagpur, but I'm also a visiting faculty to IIT Madras and a couple of other IITs. In case of IIT Madras, in 2011 or 12, they have this concept of Tech Fest, Technological Fest. It is not a song and dance jamboree, okay? This Tech Fest is a place where students from all over the India and sometimes abroad also happen to participate, and they're given different problems to solve. In 2011, I have forgotten the exact year now, maybe it was 2011, I am associated with the Department of Biotechnology and Chemistry, both in IIT Madras, where I'm a visiting faculty at every year. So they had invited me as a judge for the Tech Fest that they were holding, and the problem was up to me to give. And the problem that I've given them is exactly this: that there is no global scoring function. So a couple of very representative targets were chosen to make life simple. We chose it from the PDB database, so that one doesn't have the time for going to homology modeling and all of that. So among crystal structure available, we gave a set of targets, and then of course I will not get into what exactly was the problem that we gave, but the point is that at the end of the day, it was there for the people who are participating to realize that there is no possibility of a global scoring function, simply because for across all the known protein targets, simply because each scoring function is a sum of certain energy terms, and you cannot have a single scoring function which will encompass all the possible interactions that are there. Till now, it has not happened. So that is one of them. That's why I think I've written here, or maybe it's not there, but this is one of the problems actually. Apart from that, that this method of docking, usual method, that they rely on reasonable poses of ligand. So it is a pose, very much pose dependent, method. Yeah, it is here. No docking method for all targets. Then there's a very severe problem. You see, whenever you do docking with all the single precision, multi precision, this precision, that precision, free energy of contribution, binding with entropic terms, blah 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 blah. End of the day, and we have seen this in so many cases. End of the day, when you are trying to correlate the experimental activity with the let's say binding energy, let's say you've made it very accurate, free energy of binding with the entropy term and all that, still you will find that the correlation between, that means the most active molecule, you expect to have a very good score. It doesn't happen. Okay, my friends, it doesn't happen that way. Majority of the time, there's no direct correlation between the scoring function, whatever scoring function you have taken, it could be the binding energy also, and the activity. So what do we do in those cases? So this is one of the, that's why they're marked in blue. You can see that over there. So the reason is that I told you the scoring function is just mathematical numbers. They're not sensitive enough for relative ranking. Apart from that, the binding energy and the docking score also do not give you direct clues. Direct clues, you can indirectly do that for designing a better ligand or lead optimization. So there are some limitations. Now, the current or what? So, what are the ways that people are addressing these problems? One of the ways in which it is to obviously make the scoring function more and more rigorous. Like I told you, it will become more computationally demanding, more computational costs. You will add more terms and make your life more miserable. That is one way, but that is one of the known ways. The other way is what I told you, just like empirical scoring function. What you do is you try to statistically, that means through QSAR, use QSAR to correlate the binding. It could be either binding free energy or affinity or activity with the interaction energy terms. So you know very well these are the interaction energy terms. So it is building up statistical models, which I already told you when I was discussing empirical scoring functions. So when you do that, you actually try to bypass the limitation of the poor correlation and the activity by breaking it up into its corresponding. So this is what I call as the cause. The cause for good docking are these good interactions. So you go to the microscopic level of the interaction and try to build up statistical models. And then there is one more method which is called to address this problem. Is called scope. The full form of scope is this. This is our own method, by the way. Scope is our own method, our own innovation from V Life Sciences. And though it has a full form, according to me, scope is a method which really improves the scope of docking. And maybe just a couple of slides before I end this presentation, how it does that. So in this, one of these publications is for that scope. I will come to that in the next slide. Which one? Yeah. So here what we do is, so there are two ways in doing scope. One is either you do it from a binding energy point of view, but it is time consuming because you have to optimize the protein ligand complex and all of that. Other one is much faster, is do it through the score itself. So in the energy score, you have these different scoring terms, which are nothing but the energy of the protein and ligand. Those can be used as properties or descriptors, and you have anyway the experimental activity, and you can try to correlate. So in the first stage, what we do is we take the docking poses of the ligand, and for each of these poses, you have a corresponding score. Try to break down the score into its corresponding scoring terms, and those become the important properties or descriptors. Second level, stage 2, we try to build up a QSAR. We are very good in QSAR. We've got very good QSAR techniques in our software. So we'll use a QSAR modeling technique in order to correlate the activity of the molecule with the scoring terms. This is what is done over here. Okay. And in the process, obviously, because we have the full power of QSAR in our hand, we are able to come up with good QSAR models, which is able to reproduce the experimental activity and versus the predicted activity through a QSAR model. But mind you, this QSAR is QSAR model based on the docking results. So you can say it's a hybrid concept. It's a hybrid concept. So at the first level, we try to correlate the experimental activity with the predicted activity, which was not there in case of just the raw docking scores. But now we can have that because we are going down to the microscopic level. But beyond that, you know very well at the second level of QSAR, you can also use those information. For example, the contribution, which are more contributing descriptors than the other. You can use that particular information to develop your new molecule or lead optimization. So that's why it is marked in blue. Aid in lead optimization to improve it. I'll show that with some. See, again, this is because we use the PLP scoring function. That's why the same slide has come. I've already covered that earlier. This is the entire flow, the flow chart for scope. So for scope, the input is a complex. It could be either a protein ligand complex through docking, or you can take a co-crystal complex from the PDB site. So input for scope is either a docked complex or a co-crystallized complex taken directly from the PDB. In either case, we try to find out the residue wise. So what is important in case of scope is whatever I told you just now, in terms of the properties or the descriptors, we are trying to calculate the interaction terms or interaction energy term between the protein and the ligand as properties, but we are trying to do it residue wise. This is the key of scope. Residue wise means that it is no more why scoring function fails, because it is a global function. But when we go down, break it down to the microscopic level, and we go to the residue contribution. So for each of these scoring terms, we try to find out the residue contributions in the active site, and those individual residue contributions of the binding interactions are our actual properties. So this is our, you can say, our trump card in, not Donald Trump, who is, I think, winning. My colleague was seeing the results, and again for the second phase, Donald Trump is coming, but that's a different story. I'm saying that in case of our docking scope, I just got deviated because I used the word Trump. But anyway, trump card, the trump card in scope is this particular microscopic insight into the binding scenario rather than just a global scoring function. Now I told you why it works much better than the raw docking score. And these are the publications, by the way, our own publications when we first introduced this concept. So here, what we do is, after we break down the scoring function individual terms in terms of the active site residue contributions, we use them as properties, and experimental activity is already there. We can build up good statistical models, QSAR, and after that, we can not only use for screening or prioritizing the molecules, whichever has got better activity than the others, but also we can find out what are the key residue interactions. The key residue interaction means which are contributing more residue, which residues are contributing more than the better than the others. So in other words, if you ask me, it is a QSR model, no doubt about it. It is a QSR model of the docked or co-crystallized poses. It is able to improve the ranking or prioritizing of the ligands compared to raw docking results. It is also able to contribute, find out which key residues contribute more than the others from the contribution plot. And using all of this information, you can actually not only predict unknown compounds. So at one level, it can be used as a very good virtual screening tool, because once you have developed this, you can use it for screening large databases after you have searched molecules on the basis of pharmacophoric properties or whatever it is. So you can serve as a very good virtual screening tool. At a higher level, it can also use for lead optimization. So this is one typical, there are several case studies which are reported with this method. One of them I'm showing you. This is a typical antifungal work which we did for one of our clients, and this was typically using voriconazole. These are all antifungal molecules against Aspergillus fumigatus and Candida albicans. This is old paper, as you can see. So there are three graphs over here. Okay, so these are the molecules and these are the corresponding activities. You see, the experimental graph of the rank of activities is the blue graph. The blue graph. Obviously, these are better candidates. You can see that they are higher up in the graph. But anyway, this is the blue line with all the points are the experimental ranking in the lab activity wise. If you take only the raw binding energy as per, let's say you have done it by binding free energy, you have introduced entropy term and all that, tried to make it very, very accurate. But the plot of that raw docking score in terms of binding energy is like this. So you can see, I don't have to tell you that the prediction is horrible prediction. You can see that. Apart from maybe one, all the rest are very poorly predicted. In that context, when you take scope, you see, I have only given, it's a large data set, only representative I have shown you 8 data points. Out of 8 data points, you see 1, 2, 3, 4. At least 50% of the data points, when you take scope, and mind you, this is not the entire plot. Couple of years back, I was presenting this in IDEM. Old story I remember in Madras University, in the the Department of Crystallography. It was a conference. I was invited. One of them told me, sir, actually, why you're showing only 8 data points? I said, my dear friend, it is not 8 data points. It is more than 150 data points. But if I give all of that, it will become so jumbled. Neither you nor me can understand what I'm talking about, what you are understanding. It is like that slide I showed a little while back. Everybody is nodding their heads. Nobody understood. So the point here is that I'm just showing a representative just to keep it as uncluttered as possible. But the point is very, very clear. Scope gives us much better ranking than only the docking score in terms of binding energy. Again, So there is see that these two papers are our own papers when we launched Scope. This is some of the industry work. These are not published. These are taken from the industry work that we have done with antifungals with some pharma companies. This one is actually a very interesting publication, not from our group. This is one of our users. This is, I'm sure you know him. He's a very much active member in AICTE conferences and all that. Maybe he's there also in this conference. I don't know. I have not seen the list of all the speakers. Kunal Roy. Kunal Roy is a good friend of mine. He's from Jadavpur University, Calcutta. My home place also is Calcutta. He's one of the most leading QSR, academicians today with lots and lots and lots of publications. He has published a lot using our QSR software, our software. But apart from that, he has also used our Scope. And this is the publication of Dr. Kunal Roy and his group in Molecular Informatics 2012. Where what he did was he had a set of poses. See, the input here is a set of poses. I already told you that, right, in Scope. So it doesn't have to be always through our software. I think he had performed all these documents using, I've forgotten now, maybe Accelerate's software. And those poses were fed into our, into our Scope. And because the input for Scope is the set of poses, and then he did Scope, and he showed how nicely they matched, in terms of the experimental activity. So it's an interesting publication. That's why I've kept it on the slide, other than our own publications. Latest publication, you can understand this is 2020 running. This slide is not, I have not changed it. Those times it was latest. Now it is old, but it is interesting. The paper is interesting. If you're interested, we can share that with you. Anyway, so this is another case study with one of our industry projects which we have done. So this is regarding utilization of Scope with PTP1B. This is type 2 diabetes with one of our pharma clients. It's a big case study. I'm not getting into that because I know this will be towards the end and nobody would have the time and energy. But just showing you some interesting things that come out of this. When we do the Scope using, a set of course, a set of active molecules and, a set data set basically, which bind to PTP1B. If we get the contribution for ultimately after doing Scope, this is Scope 1, Scope, we did it a couple of times. So we find if you take one of the plots, let's say this plot, okay. So these are the different active site residues and their corresponding contributions, okay. And you can see very well that there is steric. So there are, you will see with the corresponding active site, either it will be ST or it will be HB. HB is hydrogen bond contribution, ST is steric contribution, because in PLP, these are the only two terms. I told you that. And you can see very well that some of them are contributing more and some of them are contributing less. So from this contribution plot itself, after doing Scope, you will come to know which are the residues which are contributing more and which are contributing less. However, here there are also some more take-home messages. See, these are all negative. Okay, fine. Some are more contributing. Those are more interesting to us. Some are less contributing. Okay, but some are positively contributing. So this ALA 217 from the A chain of PTP1B, both its steric and hydrogen bonding contributions are positive, and that is very bad. So these are the ones which have to be avoided. In other words, supposing I'll go ahead with the other slides, but I'm telling you when I'm on this slide, looking at this slide itself, I'll get an information, so many information I can get that what is the importance of the interaction, which is interacting more. So when I design my new, after seeing this contribution plot, if I want to design my new molecule, if I want to do some lead optimization, then I will place active molecule in my data set inside the active site, get these contributions, look at this graph, see which are the more contributing terms. For example, valine 49A chain steric interaction, very prominent. Arginine 47A chain steric. I will try to modify the functional groups on the ligand, most active molecule placed inside the active site in such a way that these interactions improve. And I will also see that since these are unfavorable interactions, they are in the positive direction. When I design my new molecule, I will try to see that these interactions are minimized, or I don't introduce groups which will capture those interactions more. So this is nothing but inputs in terms of lead optimization. Beautifully, it will come when you. But let me tell you, it might sound, it might look very nice, but it is a challenging exercise. It's a challenge. It's not an easy exercise. None of the molecular modeling work is easy. Anyway, this is a snapshot after doing Scope, where you find that this is one of the active, maybe the most active molecule in this PTP1B set. These are the active site residues, catalytic domain. And out of that, you see they are marked differently. So the favorable, I'm showing you the favorable interactions, that this ligand, maybe it is valine. Yes, you already saw that in the graph, but now it is inside the actual active site after the docking. This is what I meant. Most active molecule inside the active site, you try to see which are the more contributing residues, and we have colored it differently. Steric contribution more from valine and aspartate maybe, yeah. Hydrogen bonding, purple colored is from these residues, tyrosine and then arginine, isoleucine, and all the glycine, isoleucine. So these are the ones which are more contributing towards the hydrogen bonding, and these green ones are both hydrogen bonding and steric, both this lysine 120 and the, in other words, you are getting a very, very clear-cut information about how to design your new molecule, so that when you design your lead optimized molecule, new molecule, novel molecule, it should capture interaction with these. These are all favorable interactions I'm talking about, not unfavorable, favorable, directly from the active site. So this is again a graph, similar graph. I need not again explain to you. Again, shows very clearly that if you compare the experimental plot in this particular PTB1B case, diabetes type 2 diabetes case study, once again, the experimental plot is like this. The raw docking score is very, very off track, but the scope, you can see, scope also is not that great, but at least definitely it is better than the only the docking score. Okay, now this already I have explained to you. What is the take home message? That you get an idea about the what is the contribution, who is contributing more. Arginine 47 contributing most, and arginine 24 contributing least in terms of the contribution plot. As I told you, ALA 217 unfavorable interaction you should avoid. So that's why it gives you idea. Not only ranks at the first level, because it's a QSR, it can be used as a virtual screening tool for ranking or prioritizing the ligand when you are doing a database search, for example. At the second level, at a more advanced level, you can also use all this information for lead optimization or new molecule design. And perhaps the last slide, yes, the last slide here today is that, so if I want to summarize the scope, because I told you about docking, I told you about the limitations of docking, and now I'm at a stage where I'm talking about how to address the issues of the limitations of docking through our own method called scope. So this is the paper, by the way, of the scope 2009 paper, again from us. So as I said, it is a QSAR. It uses as properties or descriptors the residue-wise interaction to the corresponding scoring terms, scoring energy terms. It is able to identify the important residues and their contributions. It also gives you a prediction of the activities, not only of the known, but you can utilize the See, all the benefits of using a QSAR you'll get over here. So that's the advantage. That's why it's a hybrid method. It is using docking results or maybe co-crystallized complexes, but that is actually in the paradigm of structure-based drug design. But it is using actually QSAR to come up with accurate results. So that is the ligand-based drug design. So it's actually a hybrid method. Okay, now and as I said, that it also helps in lead optimization. So at the first level, screening and prioritization of compounds, just like you do it in case of QSAR. First, build up the model using the known active molecules, and then use that model. Of course, see how good the model is with all the usual QSAR metrics like R squared, Q squared, F test, all that standard error. Everything of QSAR will apply over here. And once you get confidence on the good model, use it for screening large databases. So with this, I would like to. So I have actually tried. I'd like to end my presentation. All I'm trying to say is that I have tried to take you to the basic concepts of bioinformatics through structural-based drug design, the limitations of docking, and finally, what are the different approaches people worldwide are taking in order to address the issues of docking, and what we have contributed in terms of scope to this particular addressing of the limitations of docking. Having said all of this, let me also tell you that in spite of the limitations, docking is really a very good method where you get a good feel, what do you call it? Depth and feel of the active site pocket. You can, very well, a long time back, people used to give stereo glasses. Maybe we have also given with our software. You can wear those and you can get more 3D feel while you are, studying the interactions of the ligand inside the active site. So it has its own. So basically, you should understand that every method has its strengths and weaknesses. And at the end of the day, as a good modeler, you should know what to use where. As I said right in the beginning, you should know what to use where. That means you should be able to appreciate the strengths and the weaknesses of each method and apply it appropriately to your data sets. Thank you very much. Any questions?
Thank you so much, sir. Thank you so much. It was a wonderful session. We didn't realize how the time passed. So there are many, I think, some questions from the participants. Some of the
Sure.
participants have raised their hand.
Sure.
I think Mr. Deepak
Okay. Okay.
from participants.
Yeah.
He
Can you
wants
hold
to
on for a
ask.
minute? Just hold on for a minute. Yeah.
Yeah,
Just hold on.
yes, sir. Yes, sir. No problem, sir.
Yeah. Because. Q&A hai. Do I need to shift back to the? Okay. Okay. I'm just trying to come to my. I'm trying to leave the presentation slide. That's all. That's why I'm taking little time. Yeah.
No problem, sir.
Yeah. Okay. Okay. Good. I'm visible now. I see. Right. Okay.
Yeah, yes, sir. Yes,
Yes.
sir. Yes,
Yeah.
sir.
Please come up
Yeah.
with the. Yeah. Tell me. Yeah.
Yeah, sure,
Yes,
sir.
Dr. Sami,
Yeah.
a wonderful presentation. It's never heard anybody talk on this complex topic and make it so simple that I think there must be many people who have never done this but would be loving to do this now. Thanks for this engrossing session. Thank you.
Thank you so much. Thank you so much. Yeah.
Yeah, Mr. Deepak, you can ask your question. Yeah, Deepak Penuli, sir, you can ask your question. Sir, are you there? Okay, I'll switch to the next participant.
Okay.
Yes, sir. Deepak, sir, are you there? Is there any issue with your mic, sir? Deepak, sir, you are not audible. If there is any issue with the voice, you can paste your question in WhatsApp group. I will ask the question to the speaker, sir. So you can type your question in WhatsApp group, sir. There was another participant, Miss Neelofer. She wanted to ask her a question, I think.
Okay. Yeah.
Yeah. So, ma'am, are you there? I'm not sure, sir, because she raised her hand. There might be some issue. Let us wait. I think let's see if Deepak sir is posting his question in the WhatsApp group. Meanwhile, other participants can ask the questions. Question.
Sir,
Yes.
as you said, there is no global scoring function. Which scoring function you prefer in V-Life?
Oh, I see. Okay. Yeah. So as I said that, when you do this work, you must have understood now that for a given protein ligand system that you are working on, you have to see which scoring function works the best. So for that reason, every software, including our software, gives you a set of scoring functions. And in that set of scoring functions, in our case also, there are some force field scoring functions and there are some empirical scoring functions. Out of these, actually, if you ask me, there is no such thing as the best scoring functions. For a given protein ligand system, one particular scoring function will work the best. So you need to play around. But when you do that, for example, if you are using that, there's a method that we have already in our software, we have developed, and I told you that is our own contribution, that is called GRIP, G-R-I-P, GRIP. There is no choice of scoring function. The only scoring function that is given over there is called PLP, piecewise linear. I already explained to you what is PLP. Now, in the other methods that we have given, there will be a choice of scoring function, and you need to, for your given ligand protein, you use this and see which gives you the best possible result. So you have to do it iteratively. That's the whole idea. So there is no direct best scoring functions. We have, like other softwares, we have also given a set of force field based scoring functions and empirical scoring functions. So you need to play around.
Yeah. Thank you, sir. Any other participant? I think there is one participant, Ashish Bhomik. Sir, you
Okay.
can ask your question. Ashish, sir, you can ask your question. Okay. Deepak, sir, can you connect now? I think there is some issue with the voice from the participant side.
Hello.
Ah, yeah. Hi, yes. Who is it?
Ma'am, this is Deepak.
Yeah, yeah,
Are you
audible,
able
sir.
to? Yeah, yeah. Good afternoon, sir.
Good
First of
afternoon.
all, thanks to wonderful presentation by you.
Thank you.
And in the slide, you mentioned flowchart of V scope.
Yes, yeah.
Like
Yeah.
in that slide, what I wanted to know that in the first block there is structure-based study, and in between there is residue-wise interaction via docking score.
Yes.
As far as I know in the science in related to computer science and feature extraction part, is it something related to feature extraction of or learning or
Yeah. Since anyway we are talking about QSAR over here, and when I give a lecture on QSAR a couple of months back, I was doing that again like this web-based because of the corona and all that. So I said that QSAR is interestingly a very good AI on machine-based learning technique. So
Yeah.
yes, what you are saying is true, that the feature, what happens is, I will tell you what happens is that the scoring function has got different scoring terms, and these
Yes, sir.
scoring terms, for example, if I take our own scoring function PLP, there are hydrogen bonding interaction terms, attraction, repulsion, and then the steric term. Now, this particular interaction means the interaction between the protein and the ligand, protein and ligand. Now,
Yeah.
in that, so it's like a partnership. In the partnership of the protein and ligand, we segregate the protein part. So, what is the contribution? So, for example, if and these are actually numbers. So, if you have, for example, the steric contribution as, let's say, for example, the value is minus something, let's say 20 or whatever it is. Now, this is actually a participation of the ligand as well as the active site residue, because the actual partnership or the interaction is between these two guys. So we separate it out by, yes, feature extraction, because I'm not the actual software developer who did all this, but yes, I know that it is by feature extraction, like what you were saying. Yeah.
Yes, sir. That's why I asked that in the next slide, when you told about the correlation and you represented some statistical view and some features like
Yeah.
there are some positive correlated and positively some are negatively correlated.
Yes.
My confusion is that in general feature extraction and correlation, we generally prefer to take positive correlated values only. And in what you said is you are taking the negatively correlated values as a
Yeah.
main features of your model.
Yeah. No, I will this feature extraction, what you exactly mean, it is better to talk to a software developer who was actually my previous presenter in the morning, Dr. Ganesh. So it is better that you talk to him whether it is really the feature. It is just by common sense I'm saying. But in terms of the biology and the chemistry, let me explain to you what it is. See, that graph which I showed you, it is just a cartoon representation. See, the point is that it is nothing but a contribution plot which shows to what extent each of the residue contributes to the interaction between the protein and the ligand. And there you will find that some are contributing more in the negative direction, some are contributing more in the less in the negative direction. Why negative is more important here? That's what I'm trying to explain to you. Because this is actually a docking exercise, and we are trying to find out, right in my earlier slides, you must have seen that we are trying to see what is the best possible protein ligand assembly or complex, which stabilizes. We are stabilization here. We are talking about a favorable interaction over here. So that favorable interaction is always in terms of energy. More negative, the better it is. So that's why
Okay.
when it is a positive interaction, it shows that it is an unfavorable interaction due to whether
Okay.
it is unfavorable sterics or whether unfavorable sterics or it is repulsion between donor, acceptor. We are not getting into those details. But all I'm trying to say is that energy wise, it is an interaction energy. It leads to stabilization, and that is what will actually, the cause is a favorable interaction. The effect of that is seen through a good score, but that too not always. That's why I told you there are the limitations. But this is the reason why we are looking only at the negative contributions. That is more interesting to us rather than the positive contributions.
Okay, sir. As I got your point that in the stabilization method, you need negatively correlated values more than positively correlated values, right,
Absolutely.
sir?
Absolutely.
Okay, good. Sir, thank you, sir. Thank you, sir.
Welcome. Welcome.
Okay.
Yeah. Next participant is Mr. Ashish Bhaumik. Sir,
Okay.
you can ask your question. Mr. Ashish Bhaumik, are you there? Yes, sir. You can ask your question. Sir, we are unable to hear you clearly. You are not audible, sir. Ashish, sir, we are unable to hear you. I think you can post your query in WhatsApp group. So there is some issue with the voice of the participant. I think it must be some technical issue. So if he raises the question, I'll ask that question. Otherwise, are there any participants who wants to ask any query? I think participants are done with the query, sir.
Okay.
Yeah. Over to you, Sonali.
Um, Amelia, sincere thanks to you, sir, for the sea of the wisdom and the knowledge you shared with us during this amazing lecture and explaining about the docking so beautifully, its importance, its contribution for the binding of ligands, improving ranking of ligands, and so on. And once again, thank you for giving your concern and your valuable time, sir.
Thank
Thank
you
you.
so much. Thank you so much.
Yeah. Thank you so much, sir. It was a wonderful session, sir.
Thank you so much. Well appreciated. Look forward to more.
Yeah, definitely, sir. Definitely. Thanks a lot.
Okay. Thank you so much. Bye-bye.
Yeah.
Bye.
Bye-bye. Bye, sir.
Bye.
Nice.
Bye.
Dear participants, tomorrow we begin again for our day 4 sessions sharp at 10 o'clock.